Yo, what's up, Samir?
What's up?
I can't hear you.
You hear me? Hello?
No, I can't hear you right now. Wait. Oh wait.
Hello? You
Oh,
hear
I
me?
was on mute.
You
My
hear
bad.
me?
Yeah, my bad.
What's up?
I'm
I
just.
wasn't supposed to be here, but the thing I was supposed to be here the thing I was supposed to be at instead got canceled. So I'm
Oh,
here now.
let's go. How are you doing?
Pretty good, man. How's everything?
It's good. How's the Pike Halloween thing was yesterday?
It's fire.
Fire.
Oh, the haunted house. It was really good. Yeah, we made some good money. It was a
Oh,
fundraiser. So
that's sweet.
yeah. What are we learning today?
Today's another passage from like the bio/biochem section. So it's like
Like,
kind of like a I would say like more like biology experiment. Something like that.
this is only the second one, right?
Yeah, it's just the second one.
Okay,
Hey,
nice.
Kushal, how are you doing?
Good. How are you, sir?
I'm doing good. The semester's gone by so fast. Like we have only a month left, and that too, like we have fall break too. So is Cam coming today, Samir? Do you know?
No,
Oh, she's not.
nah, she's busy.
Okay, because she signed up. So I was... all right. All right, we can go ahead and get started. Let's see. All right. Share my screen. Awesome. Okay. So welcome everyone to our second MCAT Max session. Let's see. So today we're going to be walking through a passage from the biology/biochemistry section. And so it's called the SCFA and GPR41 passage. So it's like kind of like a self-signaling passage. So before we start, like we did last week, we go with like an icebreaker. So if you could just say your name, your school, and what's your favorite Halloween candy. So
I can go first. My name is Samir Nader. I go to VCU. I'm a junior. And my favorite Halloween candy, I think I would have to say, those like small, like the sour, they're like the sour straws. There's like the red, the green, and the blue ones only, and like you like kind of like search for them in like your Halloween basket because like they're like so good. Only the sour punch like sticks or whatever they're called. Those are like the best ones.
yeah.
Yeah.
My name is Kushal. I'm a junior at BCU, and my favorite Halloween candy would probably have to be like Kit Kat.
Kit Kat, yeah. That's pretty.
My name is Haryad. I go to VCU. I'm also a junior, and my favorite Halloween candy is probably Skittles.
Skittles, yeah, that's pretty solid. I would say mine is probably like Twix. Yeah. All right, so I guess we can go ahead and get started. So before, if you were here last week, we did like a background before we went into the passage. So we're gonna go ahead and do that again, just to make sure everyone kind of knows what's going on before we get into the passage. And again, like last time, we have like questions. Okay, so this first picture at the top here, can anyone kind of tell me what type of molecule that is? Like those three molecules, what type of molecule is that?
Looks like some organic carboxylic acids.
Yeah, exactly. So this is a lipid, which is, so a carboxylic acid is a form of a lipid. And so this can be like a signaling molecule. So it sends a signal to cells. So it's how the cells communicate with each other. All right. So now that we know what that is, on the other hand, the signaling molecule actually has to bind to something. So that is called a receptor. So if you can see this red, like kind of like looped thing right here, it's within the membrane. This is called a G protein coupled receptor. So this signaling molecule, this is a different diagram, but the short chain fatty acid. So this is called a short chain fatty acid because you have a carboxyl group and then you have like only a few carbons attached to it. So it's called a short chain fatty acid. So it can bind to a receptor in a cell and send a signal, right? So, okay, now that we have that concept down, so can someone tell me what a short chain fatty acid is and what does it do?
So carboxylic acid with only a few carbons attached to it, and it binds to receptors.
Right. All right. And so what's a G protein coupled receptor? And like, what is it? What's the purpose of it?
It's a secondary messenger.
Right. Yeah. So it's a receptor, and it actually it helps produce secondary messengers. So it's the first like primary messenger. So like actually the primary messenger is the short chain fatty acid, and the receptor is called like a transducer. So it converts the primary signal to a secondary signal. So and that secondary signal can lead to changes within organs. So like for example, epinephrine can cause an increase in heart rate. So it's a signaling molecule, it binds to a receptor, it causes a change in your heart. Okay. So basically how your body is controlled is it's controlled by your nervous system. So you have a brain and a spinal cord. So that's basically what your nervous system is. So it sends signals to your organs. And how it does that is through an autonomic nervous system. So the autonomic nervous system is like an automatic nervous system. You can't control it. It just moves by itself. So you have one neuron that goes to like a column right here, where it synapses with a postganglionic neuron. So this little cell body right here, this like bulge, that's called a ganglion. So this neuron before it is called because it comes before it. And this ganglion right here is the postganglionic neuron. So this is the one that goes to the heart. So the sympathetic nervous system is like known as the fight or flight nervous system, and it increases your heart rate. Sorry. And then, so yeah. So could someone describe like the sympathetic nervous system to me? Like Haryad, are you there?
Yes, so basically, like you said, it's the fight or flight, and basically it controls like your like the main functions in a way, like your heartbeat and stuff like that.
Yeah, that's correct. Yeah, so let's see. Okay. So like we talked about, before, there's two different divisions within the autonomic, aka automatic nervous system. So I like to think of like autopilot. So like it's stuff that you're not consciously in control of. So just based on the diagram here, can someone say like what the opposite functions are between the sympathetic and parasympathetic nervous system?
Like a rest or relax.
Hmm.
And on the slide it says rest and digest.
Yeah. So like specifically in the organs, like what does one do to one organ and then the other does the opposite? Like, can you give an example? Like just the diagram's right there. So
Like vasodilation, vasoconstriction
yeah,
of blood vessels.
yeah. Uh yeah, so basically it's like the sympathetic, for example, will increase your heart rate. See, it says accelerates heartbeat. The parasympathetic, you're thinking like rest and digest. So like you're gonna have a lower heartbeat, a slower heartbeat. So and then sympathetic nervous system, let's say you're in a stressful situation, you want to dilate your pupils so you can you want more light to come in so you can get a better understanding of the situation. All right. Okay. So this is again the autonomic nervous system, and we're kind of looking at the structure of it. So the setup is that we have so can someone explain what a preganglionic and postganglionic neuron are? Haryad, if you could.
So a preganglionic neuron releases acetylcholine,
Mhm.
and postganglion releases I can't say it, but yeah, you know what I
Yeah,
mean.
so like norepinephrine,
Yes.
right? Yeah, so that's good. So that is what like so what you want to know is that the preganglionic neuron is always going to release acetylcholine. Okay. So you just want to memorize that. Whereas the postganglionic neuron, if you have the parasympathetic, it's going to release acetylcholine again. Whereas the sympathetic nervous postganglionic is going to release norepinephrine. So can someone else tell me what the difference between the parasympathetic and sympathetic nervous system is in terms of like the setup?
For parasympathetic, is the preganglionic neuron longer, and then postganglionic is shorter, and then opposite for sympathetic.
Yeah, that's correct. And then like, so what's the difference in the neurotransmitters released?
Acetylcholine for preganglionic, and then acetylcholine also for parasympathetic for postganglionic, and then norepinephrine for sympathetic.
Yeah, exactly.
I also noticed, is
Yeah.
it true that the ganglion cell is within the target organ, and is that always true for parasympathetic nerves?
So this diagram shows it in the target organ, but like it's just really close to the target organ, so it's like almost
Okay.
so yeah. So another name for norepinephrine slash epinephrine is adrenaline. So that's why the receptor for epinephrine and norepinephrine, you should know this, is adrenergic, because adrenaline, adrenergic. So adrenaline binds to an adrenergic receptor, whereas acetylcholine binds to a cholinergic receptor. So if you see the stem in the word, you can tell what that receptor is binding. So cholinergic receptor is going to bind acetylcholine. Adrenergic receptor is going to bind adrenaline. So adrenaline is norepinephrine or epinephrine. Okay.
So you're using both epinephrine and norepinephrine interchangeably here. Is that true that it will bind both? Or
Yeah. So most adrenergic receptors will bind either one. So they're only different. Like they have a slight modification that makes them different. But if you hear the word adrenergic receptor, you know it can bind either one.
okay.
Right.
Wait, do you mind like just re-summarizing the difference? Like again, just one more time for me. The postganglionic and preganglionic.
Right. So the preganglionic neuron, think like of this like yellow column as like your spinal cord. Okay. So you have one neuron that comes off your spinal cord. That's called your preganglionic neuron, right? But your preganglionic neuron can't go directly to the target organ, aka the heart, for example, by itself. It needs to meet up with another neuron before it can signal to the target organ. So this is called the postganglionic neuron. So a ganglion is the cell body of a neuron. So like a neuron has like a circle, and then it has like a branch off it. So the circle is what receives signals. The branch is what sends signals. So its synapse is on a postganglionic neuron. So at the synapse, acetylcholine, which is a neurotransmitter, which is a signal, will always and then after that acetylcholine binds to the postganglionic neuron, it's going to release norepinephrine or epinephrine on that target organ, aka your heart. So what receptor would be on your heart then? Norepinephrine or epinephrine is binding to it.
An adrenergic receptor.
And what receptor would be on a postganglionic neuron?
The acetylcholine.
Yeah.
Cholinergic.
Cholinergic, exactly. So those are the two main concepts to know for that. So let's see. Yeah. So that's it for that slide. So this
I have
slide,
a quick question
yeah.
before we move on. Do we need to, could you explain real quick? I don't know if you already went over this, but the difference between the nicotinic and muscarinic acetylcholine receptors?
Right. So yeah, it does say that on there. So those are two different types of acetylcholine receptors. So for this passage today, you don't really need to know what it is, but one of them will have like an ion channel. So when the acetylcholine binds, it'll open up a bunch of ions to come into the cell. The other one will be a G protein coupled receptor, that being muscarinic. So there's two different ways they can signal the cell, but the main thing that both of them have in common is that acetylcholine's binding to the receptor.
Okay, thank you.
That is important to keep in mind. Awesome. Okay, so this concept right here is like really, really big on the MCAT. They always like love throwing this in. So if you ever like maybe worked in a lab or like read like a research article, you know, there's like a research technique called knockout genes in mice. So you have regular mice who have all their genes functional, like they have all the proteins in their body. Then you create a knockout mice, which is where you knock out a certain gene in the mice, so they're lacking like a certain protein or a certain receptor. So what this allows you to do is it allows you to see what that receptor or protein does in the mice, because you see what happens to the mice, like what function does it lose when you knock out that gene. Then you can deduce what that receptor or protein does by knocking it out in the mice. So can someone explain like the concept of a knockout mice to me?
Yeah, so you essentially like knock out a gene of in the regular mouse, and then you observe like what functions are lost, and that tells you the function of that specific gene that was knocked out.
Yeah, exactly. All right. Another important concept. So there are two things called agonists and antagonists. So in your body, you have a bunch of cells and they have a bunch of different receptors, and you have natural stuff in your body that binds to those receptors. So an agonist and antagonist is what humans have created to mimic those molecules. So like, for example, like endorphins are the natural molecules your body produces to reduce pain. But you can synthetically make that, and those are painkillers. So painkillers would be an agonist. They can bind to the receptor and have the same exact function as, like, for example, the natural endorphins in your body. Okay. But on the other hand, you can have an antagonist, which is the opposite. It can bind to the receptor and block it from being activated. Okay. So how do we tell whether something's an agonist or antagonist? Well, a lot of times in experiments, what we can do is we can graph the response of a receptor to a different ligand. And so first we just mix it with the ligand of the receptor, right? We have the normal response. Then we mix the ligand with drug 1, for example. And we measure the response. And then once we're done with that, we mix it with another drug and we see the response. If the mixture of ligand plus drug 2 produces a lower response than the ligand alone, we can tell that drug 2 is binding to that receptor as an antagonist and is blocking the ligand from doing its function. So that's one way you can tell whether a drug is an antagonist. So Haryad, could you explain that concept to us?
Just like antagonist and agonist.
Yeah, exactly. And then like maybe how you can tell from like a graph which drug is an antagonist in the graph we have there.
So basically agonists, they activate the same receptors in your body,
Mhm.
while antagonists will block them, so there's no effect. And you can see that in a graph when you like add like a agonist, and you can see how it'll affect the receptors activation, if that makes sense.
Right. So in this graph, which one is the antagonist, drug 1 or drug 2?
It's not. Sorry, hold on.
Yeah, no problem. Take your time.
The drug, like just the first. I can't see the bottom. It's not clear, but
My bad. Let me zoom in.
so would it just be the where it's like drug 1?
So it would actually be drug 2. So think of your normal response, right? Let's say everything's working well. You have a receptor, right, on a cell. And you have so what's a ligand?
Something that binds to a receptor.
Yeah. So if you have a normal response, you're gonna have a ligand binding to a receptor. You're gonna have a normal response. So that's what this first column is right here. Then if you mix drug 1 with it and you have a similar response, you can basically tell that drug 1 isn't blocking that receptor, right? So how can you tell that, Ariane?
Sorry, my computer was glitching. Could you repeat that?
Yeah. So you have like a ligand binding to a receptor in the first column, right?
Mhm.
Then you have in the second column, you have a ligand and you have a drug, but it's producing basically the same response. Oops. It's producing basically the same response as just the ligand alone. So is the drug blocking the receptor?
No.
Okay, good. All right, but now we have ligand plus drug 2.
Mhm.
Is this bar similar to the first bar on the left, like this one and this one?
No.
Okay, so what could that drug 2 be doing to the receptor?
It could be blocking it.
Yeah, exactly. And so if it blocks the receptor, what is that type of thing called?
An antagonist.
Exactly. So which drug is the antagonist?
Drug 2.
Exactly. Yeah. So yeah, so that is a big concept to know. So let me zoom back out. Awesome. So this is a pretty short background and passage today compared to last time. So let's see. So and this thing, so you probably heard of this. You need to know your amino acids for the MCAT. So what you want to do is like you want to Google them one day, just like quiz yourself on them, try to memorize all of them. So these are the different amino acids. And so what you'll notice is, so actually, can someone tell me what an amino acid is?
Just building block of a protein.
Yeah. And so amino acids have different properties. So some of them are polar, meaning they're charged. Well, yeah. So there's charged amino acids, and there's a thing called a polar amino acid, which is like a partial charge. and then you have hydrophobic amino acids. So what you want to know is that the inside and outside of cells in your body have a ton of water in it. So if you have a ton of water, do you want a polar amino acid in it or a non-polar amino acid?
Polar.
Yeah, exactly. So in your cell, what separates the inside and outside is a hydrophobic membrane. So if you have a hydrophobic membrane, what do you want? What type of amino acid do you want there?
Polar.
Yeah, non-polar, exactly. All right, so that's what we need to know going into the passage. So does anyone have any questions on this background PowerPoint? It can be any question on any of the slides.
I do have a
Sure.
question. Can you go to the receptor one? The competition. Yeah, is it I don't know if this is important for the one that we're gonna be doing, but what are the two types of inhibition? It's like allosteric and like like the competitive one. Do we need to know that for this one? But like
So
if not,
for
just
this
like a
one, you don't
refresher
need to
on
know
that.
that, but that is an important concept to know. So I can explain it for you. So competitive inhibition is where the drug kind of like competes to bind to the receptor. So it's like any other molecule that's trying to bind to the receptor and activate it at its active site. So an active site is what binds a ligand, right? So that competitor is just competing to bind to the active site. So that's competitive, right? The name is competitive competition. All right. An allosteric is a drug that binds to a site that is not the active site. So sometimes it can bind to another site on the enzyme, for example, and it can block the enzyme from working. So that is the difference between allosteric and competitive, but both of them limit the enzyme.
All right. Thank you.
Yeah, no problem. Any other questions? Okay. So let's go straight into the passage. Okay. Awesome. Can everyone see the screen, see the passage? Okay, great. So can someone read paragraph 1 for me?
I got it. Lipids and proteins are not only essential nutrients, but also act as signaling factors for various cellular processes. For instance, short-chain fatty acids, SCFAs, stimulate the G protein coupled receptors GPR41 and GPR43, whereas ketone bodies exert an antagonistic effect on GPR41.
Right. Awesome. So what are the main molecules being talked about right here?
Short-chain fatty acids
Right.
and ketone bodies.
Yeah. So what do the short chain fatty acids bind to?
GPR41 and GPR43.
Right. And so what are those things?
Metabotropic receptors.
Yeah. So yeah, G protein coupler receptors. Awesome. Yeah. So can someone else read paragraph 2?
I got it. Researchers have long been interested in the effect of such metabolic intermediates on measures of cardiac health. To test this, researchers generated mice lacking GPR41 and GPR43 and measured their heart rate, a marker of sympathetic tone. In response to the SCFA propionate, in addition to the results shown in Figure 1, the hearts of GPR41 negative mice, but not GPR43 mice, exhibited a reduction in the expression of tyrosine hydroxylase, TH. TH catalyzes the conversion of L-tyrosine to L-dopa, the precursor to dopamine, a molecule which positively affects myocardial contractility by increasing electrical conductivity in the heart.
Awesome. So it's kind of complex, so let's break it down a little bit. So what organ are the researchers kind of looking at here? What are they trying to see? So like which organ are they trying to see the effects of short chain fatty acids on?
The heart.
The heart, right? Because it says right here, they're trying to see the effects of such metabolic intermediates, which is short chain fatty acids, on measures of cardiac health. So cardiac means the heart. All right. So
Mhm.
we know what organ they're trying to affect, right? It's the heart. So to test this, they generated mice. So can someone tell me what the mice are lacking? Like what receptors are they lacking?
Just like,
GPR41 and GPR43. My
Yeah.
bad, I didn't mean to interrupt.
oh, you're good, bro.
Yeah, exactly. So they're trying to see how those receptors affect what marker?
Heart rate.
Yeah, heart rate. In response to what's our main molecule here?
SCFA propionate.
Right. So just based on that, what's our ligand and what's our receptor that the experimenters are studying?
Ligand is SCFA propionate, and receptors are GPR41 and GPR43.
Okay, good. So you want to keep that in the back of your mind. Awesome. So why would we want to knock out GPR41 and GPR43? Like what's the purpose of doing that?
To measure its
To
effect
see
on
What were you saying? Who wants to go?
I was just gonna say to see the effect of what the SFCA ligand has on like the biological function of the heart.
Right. So, but we're trying to see which receptor is responsible for impacting the heart, right? That's why we knock out the receptor, right? So here it says that, so in the graph, they graphed the heart rate versus time for the different groups of mice, but it says on top of that, the hearts of so whenever you see a minus/minus sign, that's like a symbol for knockout. So they've knocked out GPR41.
Oh, that's good to know.
Yeah. So it means that those mice had a reduction in the expression of tyrosine hydroxylase. So can someone tell me what tyrosine hydroxylase does?
It catalyzes the conversion of L-tyrosine to L-dopa,
Mm,
which is the precursor to dopamine.
right. So if you have a reduction in tyrosine hydroxylase, how does that affect dopamine?
Decreases.
Right. And so what does dopamine do based on the passage?
Increases
Positively.
electrical conductivity.
Right. Electrical conductivity, your heart can pump blood. Again, like your heart rate goes up. Okay. So if you have less dopamine in the GPR41 knockout mice, what happens to your heart rate?
Goes down.
Goes down.
Exactly. Awesome. So we know GPR41 is kind of related to heart rate. So we know it's kind of important. So let's look at the graph right here. So the big thing on the MCAT is when you're reading graphs, the star sign means that there is a statistically significant difference. Okay. So what is being measured on the y-axis right here?
heart rate.
Right. And specifically delta heart rate. So we're looking at change in heart rate. Right. So, and how you know this is you can't have a negative heart rate. They wouldn't put negative 35 right here. So we're looking at the change in heart rate at different points in time. So the x-axis is time. So we have our wild type mice, right? If you look at the legend right here, these are the white circles. They have an increase in heart rate at every single at most of the time points. So as the time points increase, we're having an increase in the delta heart rate. So we're having an increase in the change in heart rate over time, right? let's look at these mice over here, the GPR43 knockout mice. These mice have a really similar increase in heart rate over time, right? The GPR41 knockout mice, these mice, you can see right here, the line is like pretty flat, right? It's around 0. So over time, there's no increase in heart rate, right? And that's when you knock out the GPR43 minus receptor. GPR41 minus receptor. So so they're measuring the change in heart rate in response to what ligand? You mentioned a ligand earlier.
SCFA,
Propionate.
propionate.
Propionate, exactly. So propionate binds to one of the G protein coupled receptors to increase heart rate, right? So if we knocked out GPR43 receptor in mice, and we still have the increase in heart rate that happens when you have wild type mice, what does that tell you about the GPR43 minus receptor? Is it important or is it not important? If we can just take it out and the same thing still happens,
Not important.
right? Does everyone get that? Okay.
Yeah.
Whereas if we take out the GPR41 receptor, we have no increase in heart rate, right? Our line is like constant. It's just around 0. We don't have the same increase in heart rate. So does that tell you that GPR41 is important or it's not important?
Very important.
Right. So we can tell that propionate is most likely binding to that GPR41 receptor, right? Because if it wasn't binding to that receptor, we could take it out and nothing would happen, right? The same thing would still happen. You would have a graph just like your wild type graph, but you don't. So we can tell that propionate is binding to GPR41 minus receptor. And that matches, like, remember the sentence right here? It says the hearts of the GPR41 knockout mice, they also have a reduction in tyrosine hydroxylase, right? So these mice, in addition to no increase in heart rate, they also have less dopamine, right? So that's consistent. Makes sense, right? Okay, awesome. And the last part of the passage, or second half of the passage, could someone read this last paragraph?
I got it. Researchers wanted to clarify whether propionate acts through an adrenergic receptor, thereby promoting sympathetic activity, or a cholinergic receptor, promoting parasympathetic activity. After pre-treating mice with propionate, wild-type mice were then given a dose of either hexamethinium, a cholinergic receptor antagonist, or propranolol, a beta adrenergic receptor antagonist, which is structurally similar to propionate.
Awesome. So could someone tell me what the researchers are trying to figure out?
They're trying to figure out whether propionate acts through an adrenergic receptor or a cholinergic receptor.
Yep, exactly. So how they're doing it is they're mixing it with 2 different types of antagonists. So first they're mixing it with a cholinergic antagonist, right? So for example, hexamethonium, it binds to the cholinergic receptor. So if propionate was indeed binding to cholinergic receptor, if you mix it with hexamethonium, you're going to have a lower response, right? Because it's going to block the receptor. But we don't know if propionate binds to that receptor or not, right? So that's why we're doing the experiment. So propranolol is a beta adrenergic receptor antagonist. So this one blocks the beta adrenergic receptor. So we're using these antagonists to figure out which receptor propionate normally binds to. Because if the antagonist, when mixed with propionate, causes a reduced response, we know that propionate is usually binding to the receptor that the antagonist binds to. Because if the propionate didn't bind to that receptor, the antagonist would have no effect, right? It wouldn't matter if, like, let's say you have a receptor X and you think the drug binds to the receptor X, but you mix it with an antagonist to receptor X and there's no reduction in response, that would mean the drug's not even binding to receptor X. So that's it's a bit confusing to think of, but let's kind of look at the graph here. So again, what are we measuring on the y-axis?
Heart rate.
heart rate, right? And delta heart rate. So keep that in mind. And what do these plus and minus signs mean over here on the x-axis?
The administration of each like drug.
Right. So in the first column, what do we have?
Just propionate.
Right. And propionate is, again, what is it called?
It's the short chain fatty acid.
Right. It's our ligand. It's the normal
Ligand.
binds to the receptor. Right. Then in column 2 over here, we're mixing it with which drug?
The cholinergic.
Hexamethonium.
Yeah, the cholinergic one, hexamethonium. So hexamethonium binds to the cholinergic receptor, right? Okay, and column 3, what are we mixing together?
Propionate with the propanol.
Exactly. And what does propanolol bind to?
Adrenergic, which is parasympathetic. Sympathetic.
Exactly. So we're trying to figure out which receptor does propionate normally bind to, because if you mix it with an antagonist that binds to that receptor, you should have a reduced response. Okay, so based on column 2, what can you kind of get from that? Like, what is that telling you?
I mean, it looks like hexamethonium isn't competing with propionate for the receptor, meaning that it's not the parasympathetic cholinergic one.
Right, exactly. So we know propionate does not bind to the cholinergic receptor, because if you mix it with hexamethonium, a normal antagonist for that receptor, it doesn't change the response at all. So it can't be that propionate's binding to that receptor. Okay. Now either Krishal or Andy, could you explain what the third column tells us?
When you add propanolol, the heart rate goes down, so it must be an antagonist. So propionate acts through an adrenergic receptor.
Exactly. So does everyone get that? Awesome. Okay, now let's move on to the questions. And again, we have a star sign right here. Star right here, and the star is comparing the normal propionate to propionate mixed with propanolol. So that means this difference is significant. All right. So question 1, what is the structure of L-DOPA? So where is L-DOPA mentioned in the passage?
The second paragraph.
Right. What does it say about L-DOPA?
The precursor to dopamine.
Okay. So can someone explain how to answer this question?
Do we have to know its structure or dopamine structure?
Yeah. One second. I was actually on question 4, so keep that in the back of your mind for later. But question 1. So can someone read question 1?
In the hearts of mice lacking GPR41, the concentration of which of the following is most likely reduced?
Okay. So it says here that in addition to the results shown in figure 1, the hearts of the GPR41 knockout mice have lower tyrosine hydroxylase expression. So what does tyrosine hydroxylase do?
Catalyzes the conversion of L-tyrosine to L-DOPA, which is the precursor to dopamine.
Awesome. So what should happen to, so if you have mice that are lacking that enzyme, which of the following is going to be increased? What are you going to have more of?
Tyrosine,
Right. And why is that?
because it won't be converted.
Yeah, it won't be converted to L-DOPA, and it won't be converted to dopamine eventually. And propionate is not even related. So awesome.
I have a question. Hold on. So I understand why it's A. Like that's like the obviously like the best answer, But couldn't it technically also be D, because now that there isn't a receptor, there's more of the ligand?
Let's see.
It would be down regulated though, I'm pretty sure in that case. No?
Well,
You're saying just like the body would make like less of it?
so dopamine and the short chain fatty acids are both ligands. So they're both stuff that binds to the receptor. So they're like different ligands that can bind to a receptor, but they're not really like related in that case. Does that make sense? Like they don't really
Yeah.
have any effect on one another. But the reason why the answer choice is there is because it's for people who didn't understand the passage or are just looking for words in the passage that seem familiar. So that's kind of like a test maker insight. So, but we know from here, it says we have like, so that's a good point, Samir, though, because like there can seem like there are multiple right answers, but you want to go with the answer that has like the most evidence, the one that you're the most confident about, even if there seem like there's multiple right ones. So here we have direct evidence that they're going to have less tyrosine hydroxylase, less dopamine. So
Logically, would there be any reason for propionate to have change?
not that I can think of right now.
Okay.
I would say yes, because if you're missing the receptor, then you have more of the, like you have like a
More like
backup
propionate.
of the ligand. Yeah. You know what I mean?
Yeah.
Okay.
That could make sense if like you think about it, but like,
It's
they
definitely
want
not
you
as likely.
Yeah,
Yeah.
they want to push you in a certain direction based on the passage. So you don't want to get tied up in that. So that's that. But good thinking though. All right. Which amino acid is most likely found in the cytoplasmic domain of GPR41? So cytoplasm is what is that?
Polar.
Yeah, it's the inside of a cell. What did we say about the inside and outside of cells in the PowerPoint?
They're polar.
Contain a lot of water. So
Exactly. They have water, which is polar. So if we have a watery polar environment, what type of amino acid is going to be in that?
A polar
serine,
charged one.
sorry.
Yeah, exactly. A polar amino acid or a charged one. So does anyone here know what these amino acids are? If not, we can just like pull up the chart.
The first three are all non-polar, and the last one's polar.
Yep, that's right. And I'll just pull up the chart. Just 1 second.
Wait, can you explain that again? If we're looking at the cytoplasmic domain, why do we want a polar one?
So the cytoplasm always has water inside of it.
Yeah.
So water is polar. So
Yeah.
polar likes polar. So that's why we have a polar.
Okay.
So he quickly pointed out that so the first three options I believe were like alanine, methionine. So these are all the hydrophobic ones. So again, it's a good thing to memorize your amino acids. So we see these are hydrophobic. So what you're looking for is the side chain. So all amino acids have a carboxyl group and they all have an amino group, the nitrogen. But what you're looking at is the different side chains right here, right? So these are all non-polar, okay? And what we have though is one of the options was serine. You have a hydroxyl group right here, right? That is polar. So it's gonna like water. So we would expect to find it in the watery cytoplasm. Does that make sense? And again, just good thing, just like memorize all your amino acids and know which ones are charged, which ones are polar, which ones are hydrophobic. So it's a good thing to do. Okay, let's go back to the passage. Okay, so he said serine, so that's why it's polar. So we find cytoplasm, and that is correct. Awesome. Okay, and this is requiring you to read the graph. So which conclusion is best supported by the data in figure 1? So take a second to read that, and if someone has an idea on how to answer the question, feel free to speak up.
Do you want to answer or just like?
Yeah, sure. Like you can give the answer and then just like say how you got to it.
I would say the answer is B, because that's what the graph shows, that when there's the GPR43 is removed, it still had an increase. But when GPR41 was removed, then it wasn't.
Yeah, that's exactly correct. So yeah, so B is the correct answer. Does anyone have any questions about that? Is anyone tripped up by the other answer choices? Want me to explain them?
Can you talk about
What is
D?
D saying?
Yeah,
Yeah.
I was just about to ask about D too.
So D is saying that GPR41 is responsible for cellular uptake. But so GPR41 does respond, it responds to the short chain fatty acid, aka propionate, but there's no evidence that it actually causes its uptake into the cell. If I go back to the PowerPoint, you see how we have a G protein coupler receptor? It's just binding to the outside, right? It's not actually going inside the cell, right? It's not being uptaken into the cell. So the receptor isn't taking it into the cell. It's just interacting with it on the outside. So there is no cellular uptake, right?
That's just a trick answer.
Yeah. Does that make sense? If it doesn't, just feel free to speak up. Okay. Okay, and that is the correct answer. Okay, back to question 4 where we started. So L-DOPA is made from L-tyrosine. And so what happens to L-tyrosine, Krishal? What does tyrosine hydroxylase do to L-tyrosine?
It adds a hydroxyl group, rho H.
Yeah. So if we know the structure of tyrosine, how can we figure out the structure of L-DOPA?
Just by adding a OH group.
Exactly. Okay. So stay with me here. Awesome. Okay. This is the structure of tyrosine. Okay. Everyone remembers that? So like, okay. Okay, so we know the structure of tyrosine, right? We want to add an extra OH group here. So Krishal, so which one is going to be the answer?
It's going to be A.
Yep, exactly. Yep, and that's correct.
One booklet.
Does that make sense to everyone? Awesome.
Yeah, because the hydroxylase is getting rid of the OH, so you need the one that has. What is the hydroxylase doing again? Sorry.
It's adding an OH.
Adding an OH. Okay.
Yeah. Right. Yeah. So that's why I show the structure of tyrosine, and we just add an OH to it. Okay. Awesome. Okay. Next question. Yeah. Okay. So can someone read the last question? All right, Alex.
Based on figure 2, propionate can be classified as which type of signaling molecule?
Okay, so based on this graph, what does propionate bind to?
Beta adrenergic receptor.
Yeah, exactly. So we kind of talked about it before when we were going through the passage that we could tell that if you mix propionate with a beta adrenergic antagonist, you're gonna have a reduced response. So the only way this could make sense is if propionate binds that major adrenergic receptor. Okay. So propionate is activating that major adrenergic receptor. So is it an agonist or antagonist?
Agonist.
Agonist. So it would have to be beta adrenoreceptor agonist. And that's the correct answer. So let me stop sharing. Does anyone have any questions?
Yeah, I do. How do you study, like, what's the fastest way to learn all the amino acids? Because I missed two of the questions because of like I would have missed the structure one because I don't know what L-tyrosine looks like,
Yeah.
and the other one because I didn't know which ones
So
are
the
polar
first
or
way
non-polar.
is so Google amino acids, right? Look at a chart of them. Like do a quick scan of them. Once you're done with that, get a blank sheet of paper out and write all them from memory. Like try to. Like you're obviously not going to be able to recall most of them, but it's a good idea for making that information stick in your mind. So try to recall as many as you can. When you can't recall the rest, look back at the chart, do another scan, close your computer, go back to the white sheet of paper, and keep writing out the rest of the amino acids. And then once you're done with that,
Do you need
do it a whole nother time. Except don't look at the chart. Just like try to write all of them out from memory.
do you need to know the structure of every single one too?
Yes, you need to know the structures of all of them and all the classes of them too. Like they're pretty picky about them.
Okay.
Yeah.
Interesting.
When you start to do it, you'll start to notice patterns too between them that'll help you memorize.
Yeah.
There's
Like
also-
within their
Yeah.
groupings and stuff. And there's also an app you can use later to
Yeah, that's what I was gonna say.
solidify your knowledge. Yeah, there's a quiz, amino acid quiz app, which is kind of fun for timing yourself. And
That's pretty helpful.
yeah, it makes them like
I need to
second nature.
get that.
Yeah. So any other questions with the passage or PowerPoint?
Is this the app?
Yeah, that's it.
Yeah,
This is the one. All right. Good looks. Thank you.
Yeah. That should be great for memorizing them.
Sweet.
So I guess if no one has any questions, does anyone have any feedback about like how we can make this better?
I thought it was pretty good. I like how you do the review of the concepts, like the content review before we head into the passage, because it helps, it like refreshes our jogs our memory. That way we don't have to go back and forth.
Yeah.
Oh, I have a question. So would you say that it's always best to approach these questions, I guess, by reading the passage first, or do you ever like read the question and then go back to the passage? Because
I
some of
would
the questions are sort of unrelated to the passage.
you could do that, but sometimes you can get like a bit confused if you just try to answer the questions without the passage. Like you might really have context. So I always find it best to read the passage before the questions.
What about if we have issues with like time? So right, because like obviously when we review them together right now, we're not really concerned about time. We're just focused on understanding the concepts and getting the questions right. But on the real test day, there's a time crunch. So do you have
Yeah.
any like time saving strategies you could share with us?
So what I would say is my personal strategy is to try to read the passage as fast as possible and try to make sure try to make sure you understand the passage, and then try to go through the questions with like your like first instinct. Because what I find is if you try to read the questions first, you'll constantly be glancing between the questions and the passage, and that could like chew up a ton of time that you could have saved if you just got a good understanding of the passage the first time around. But everyone is pretty different, so it's like you have to try to like experiment with yourself and try different strategies. But my personal advice would be try to get a good understanding of the passage, understand these graphs, then attack the questions. But everyone is different.
kind of building off that, do you do you usually like, when you're reading the passage, do you kind of like read a paragraph and then try to understand the paragraph? Like that's what we did today.
Yeah.
Do you usually do that, or would you just
Yeah.
like read the whole thing?
So yeah, so I would read it and try to understand it mentally. Like, so these questions that we're doing right now, you want to have them like do those mentally, like quiz yourself in your mind. What is it talking about here, right? These questions are a whole lot easier to answer when you understand the passage. Whereas if you don't understand the passage, don't know what's going on, all this is gonna seem like jargon. You know, you're gonna have a tough time. So yeah, that's my personal strategy, I would say.
Thank you.
Anything else? So I'll stop sharing. So our next session should go back to like Mondays. I didn't have it Monday this week because it was Halloween. So it should be next Monday should be our next session, and we should have our people from last week back, hopefully, and more people. So make sure to spread the website to as like many people as you can if you ever run into like people studying for the MCAT, or if you know anyone like that. So yeah, that's my takeaway, and we'll have all this up on the. So like, did you guys see the website last time? It was like updated with the video of our last session, and all the PowerPoint Word docs are on it.
Yes.
Yeah. So this one will be up there on it as well, so you can go back and look at it. And yeah, that's pretty much it. So everyone's free to go.
Sweet. Thanks, Sid. That was super helpful.
Thank you so much.
Thank you.
Yeah, no
Thanks.
problem.
Bye, guys.